Hello to 13.2. I'm going to post some links under this video because halfway through the video I'm going to have you watch those instead. I'd watch them together on this, but then that makes this video crazy long and it takes forever to upload and my computer might not be able to handle it, so I'm just going to post the links to those stuff and then you guys will have to watch those in a little bit. So we're talking about genetic technology, and 13.2 is titled Recombinant DNA Technology. So here we have a tobacco plant that contains a gene from a firefly. It is glow in the dark. We literally took genes, DNA out of the firefly and put them into a tobacco plant. And now it can glow. It'll still be a tobacco plant. It'll still do its own thing. But it'll also glow in the dark, which is kind of crazy. So we're going to learn how we do that. So the applied genetics that we learned in 13.1, the selective breeding, yes, I guess that's kind of a type of genetic engineering, but the real definition of genetic engineering is a faster, more reliable method for increasing the frequency of a specific allele in a population. And what we have to do for this is cut some DNA or cleave some DNA from one organism into a bunch of small pieces and inserting the fragments of that into a new organism. And that new organism can be the same species or a different species. They don't have to be the same. Remember, all living organisms are still based off of the four letters DNA, the A, T, C, and G, right? So you can put DNA from another species into another species that aren't anything closely related because we're all still built on the same four letters. So as long as you can find a correct spot to put it in, and we'll see what that looks like, then you can do that. You may also hear genetic engineering referred to as recombinant DNA technology, because we're recombining, so recombinant, that's what we get that word for. If we can put DNA into a new organism, then that new host organism will use the foreign DNA as if it were its own, because remember, your body's just going to read the instructions, and if you come in and change the instructions, your body's still going to read it. It doesn't matter where the instructions came from. If this happens, those organisms are known as transgenic organisms because they contain foreign DNA. So this is a transgenic organism because it has foreign DNA inside of the plant. It has the firefly stuff inside of the plant. So there's, three steps? First, isolate the foreign DNA fragment that will be inserted. So in the case of that glow-in-the-dark plant, let's isolate the DNA from the firefly, or dragonfly, or whatever it was. Firefly, yeah, duh. Dragonflies don't light up, fireflies do. Sorry. So isolate the firefly DNA. Second step, attach that DNA to a carrier. And then third step, transfer that into the host organism. Okay? So isolate the DNA, attach it to a carrier, and transfer that into the new organism. Those are the three steps. In the 70s, we found something called restriction enzymes, right here. And these are bacterial proteins that have the ability to cut both strands of DNA at a specific sequence. There's a bunch of different types of them. This is just an example of one. It gives the name right here. This thing goes in and cuts it at a very specific spot. So if we can isolate a piece of DNA that has the same kind of plugs that the original DNA has, then we can put this chunk of DNA and insert it into the other DNA. And it's thanks to those restriction enzymes that are able to cut the DNA at specific points that we're able to do that. Notice that this is AATT, and right here this is AATT. So we're pretty much just adding this extra CG in the middle of our regular DNA strand. Your body's still going to read that and still produce some stuff, but now once it reads this part, it's going to produce different amino acids which end up making different proteins, right? Like maybe the glow-in-the-dark protein that we were looking at for the tobacco plant. This isn't the only way you can cut. This is just one example. Here we see you can cut blunt ends sometimes. You can cut palindrome sequences, which means they're the same back and forth. You can unzip it for a few nucleotides. You can do all sorts of different. Up here it says there's hundreds of them, right? So They have a couple different methods of being able to do this, but the idea is the same. We cut at a specific spot, we insert a matching plugs but different information on the interior, and now this piece of DNA has this foreign DNA in it, but it fits just right with the instructions to where your body won't be able to tell, or the tobacco plant's body won't be able to tell in the case of this glowing firefly thing. Now, in order to put that foreign DNA into a host DNA, we have to use what's called a vector. A vector is the means by which DNA from another species can be carried into the host cell. So we got to attach our little free-floating pieces of information, attach it to the carrier or the vector in order to get into the other organism. There's biological ways to do this, but there's also mechanical ways to do this. One of the biological ways is a virus or a plasmid. So both of these can enter an organism and change the DNA. Viruses we will go over next chapter, but a plasmid is like this little dude, small rings of DNA in bacteria. The genes it carries are different from those on the larger bacterial chromosome, but if we can throw it in there, then the bacteria can still read these instructions, and we'll see what that looks like in just a little bit. So those are two biological vectors, right? Viruses can enter a cell and replace DNA. That's what viruses do. And plasmids in bacteria can read instructions of outside of their main chromosome. Okay? So viruses go in, change the DNA. Plasmids are these extra little DNA things that bacteria can read. Another vector, we said, were mechanical vectors. And that comes down to stuff that's man-made. We made it. So one of the examples is a micropipette, which is just inserted into a cell and pretty much shoots all the injectable DNA into a cell. And another way, which I tried to find a cool video but I could not, is a bullet coated with DNA that is shot into the cell from a gene gun. So if you thought biology was boring, just go study that and tell me what that looks like, because that sounds pretty awesome. Alright, so as we've learned, if a plasmid and foreign DNA have been cleaved with the same restriction enzyme, the ends of each will match and they will join together, reconnecting the plasmid ring. The foreign DNA is recombined into a plasmid or viral DNA with the help of a second enzyme, and we will look at that later. So what that means is there's a second enzyme. The restriction enzyme cuts the stuff, and then there's another enzyme that puts it back together. After the foreign DNA has been inserted into the plasmid, the recombined DNA is transferred into a bacterial cell. So that's what's happening in this picture up here. Once you recombine the plasmid, you put it back into the cell. The plasmid is capable of replicating separately from the bacterial host and can produce up to 500 copies per cell, which means we throw one of these guys in the bacterial cell, and this guy on his own, essentially, will make a bunch of copies inside of this one DNA, or inside of this one bacteria. Now, bacteria reproduce really fast, so while that's happening, the bacteria is also reproducing with this new little plasmid ring in it, right? So now we have a million different little bacterias, each with our changed plasmid inside of it that's making some weird new compound that we injected into its DNA. Now the word clone, in case we're not sure, means genetically identical copies, and each identical recombinant DNA molecule is called a gene clone. So here we see these are gene clones. Plasmids can also be used to deliver genes to animals or plant cells. Each time the host cell divides, it copies the recombinant DNA along with its own. So if we throw a plasmid into an animal or plant cell, when that cell replicates, it will also replicate these plasmids. And then our cells can read those, or animal cells can read those, and produce the proteins that are now written on this extra piece of instruction that we have thrown into a cell. So here we see a plasmid. We cut this. We insert foreign DNA, so this is bacteria, but we're putting human DNA into it, so it makes the human growth hormone. So now this is bacteria, but all the red is human genes. We throw this plasmid into a cell. This little bacteria makes more copies of itself, and the genes itself makes copies of itself, and now this bacteria is making human hormones that we can then like pick up and then use as a medicine, or as a like a medicine or a treatment or something like that. So it's pretty crazy stuff. This book's kind of old, so it's saying cloning of animals. This will say like, oh, we're almost there, we can almost do it. We can definitely do it now. We can definitely clone animals. We can probably clone a person, but nobody's going to publicly do it because that's kind of unethical, we think. And I know second period, we've talked about that before. Like, if a company makes a clone, then does is the person a person, or does the company own the person, kind of thing like that. So there's all sorts of crazy ethical problems involved with cloning people. But most people don't see a problem with cloning animals. An ethical problem with cloning animals, at least. What happens, though, is if you clone a 10-year-old sheep, then the new baby sheep that you cloned, its genes are already 10 years old, so it starts developing health problems way quicker because it thinks it's 10 years old, right? Even though the baby is new, the genes are 10 years old, so certain things start breaking down way earlier. At least that was the case with this particular animal, Dolly, the first cloned animal. So for PCR and for sequencing DNA, I'm going to post two more video links under this video. So what you should do right now is pause it, watch the PCR video, watch the sequencing DNA video, and then come back to here. So go ahead and pause it and then come back. Alright, this is assuming you watched these two videos. Now we're going to continue with just this last bit of information. Applications like why would we do this, right? We can do this, we can clone animals, we can insert genes into other stuff. Why would we do this? Well, one of the ways, let's look in industry, is we've modified a bacteria called E. coli to produce indigo dye. Okay, so indigo is a blue dye. It's pretty rare, but if we can just inject the instructions to do that inside of bacteria, then make it for us. We don't have to get it from wherever we get it from normally, which is a very cheap alternative. So industry has use for this because they can say, you know, this compound is important in making your clothes. How can we get more of it without paying as much? Well, let's find out what the genes are. Let's make a new bacteria to be able to make this compound, and then we don't have to, you know, go out and find it. We can just make it in a lab. So that's why industry might want it. Let's look at medicine. So one of the big deals that we can do this is insulin. So people with diabetes don't have any insulin. You might think, well, let's just take it from people who do. And that's a decent idea, but that would be very not, it'd be very time consuming. So instead what we can do is, let's take the gene for making insulin, throw it into bacteria, and then let's see if that can produce insulin. And it can. Right? So we take your genes that produce insulin, throw them into bacteria, the bacteria then produces insulin, and now we can give that insulin to people who can't make it, and now they're healthy. We can do this with antibodies, hormones, vaccines, enzymes, all sorts of crazy compounds. If there's something that the human body can make, then we can just cut those instructions out, put it inside of a DNA, or inside of a bacteria, and try to make that bacteria make it so we don't have to harvest it from people. And then we can give it to the people who need that kind of stuff, who might be, their hormones might be imbalanced, or they don't have insulin, or they need a specific enzyme because they can't function without it. Another way we use it is to study the way diseases spread or the way organisms develop. A friend of mine actually has spent the last 10 years using a fruit fly. And what he does is he goes in and he messes with the DNA a little bit and makes certain things glow in the dark, and sees where like the nerve cells connect and where the nerve cells trigger because of certain smells and stuff like that. So that's useful because if we know how simpler organisms function, then we can take it a step up and go to more complex and more complex stuff, eventually being able to map out everything about the human that we can, or a dog or something, like more complex animals than just this stuff. So just being able to do this helps us understand more about genetics. Do you see that loop? We know how to do this because we know about genetics, which helps us learn more about genetics, which helps us learn more about genetics. So it's this loop that just keeps going around, thanks to the techniques that we learn from just doing crazy science like this. This is like full-on mad science type stuff, right? Like glow-in-the-dark fish and glow-in-the-dark bugs, like just because we can. We can even, in this paragraph, it explains how we can genetically modify maybe cows or sheep to produce certain proteins in their milk that humans need, almost like a sort of medicine milk, right? Like this milk now has this certain protein that people need. So now this milk is almost like a medicine for some people. We can do that. We 100% can do that, which is insane. We can make medicine milk out of sheep. Like we just mess with their DNA a little bit, throw in some certain compounds, or certain DNA, and now they can make certain compounds. We can do this with plants. We want a plant to make a certain toxin to help kill bacteria. We can do that. We get potatoes, we go inside, mess with their DNA a bit, put a little thing that says, hey, make this toxin that kills bacteria but not people, and now bacteria can do that. Hey, we want this strain of plant to be resistant to a certain type of pest control herbicide or whatever, a chemical that kills other plants. Well, let's insert the gene into it that makes it resistant to that, and then now this plant will not get killed by that particular chemical. Or let's say there's a certain area of the world that doesn't have enough vitamin C. Okay, well, let's take a plant that doesn't normally make vitamin C. Let's throw some other DNA inside of that plant, and now they can grow this new plant that normally doesn't have any vitamin C. We have injected the instructions for making vitamin C into that plant, so now those people can eat that plant and get the vitamin C that they need. Does that make sense? And anything that you might be missing, we can kind of like take it from some other organism, take those instructions from some other organism, and place it into a new organism. And I think that's about it for 13.2.